Good morning, all. The topic that we are going to discuss today is excitation, contraction, coupling. So, in this, the process of excitation and the process of contraction, and how the nerve fiber is communicating a muscle, how it is responsible for the contraction of the muscle, is going to see in this lecture. So, excitation of the nerve fiber and what is the relation between the excitation of the nerve fibers and the contraction of the muscle fiber. Let's see with the help of a diagram. So in this diagram, we can see a nerve fiber that from the cell body, it is receiving the signals, receiving the sensation, and from the axon hillock, actually the action potential started in the axon, and the action potential traveling throughout the axon, and at the nerve terminal, when it reaches the nerve terminal, it is exciting the next neurons, and it is exciting the muscle fibers, different muscle fibers. It can be skeletal muscle, it can be smooth muscle and cardiac muscle. But today in this discussion, we are continuing with this skeletal muscle. So here, when the nerve fiber is stimulated, the nerve fiber is stimulated when a threshold stimulus is applied to any single nerve fiber, then it is activated. That means the action potential is generated within the nerve fiber, and action potential travels throughout the axon, and when it reaches to the terminal buttons, the end of the nerve fibers, what happens is it causes the release of the neurotransmitters. So the main communication of nerve to nerve and nerve to muscle is mainly neurotransmitter. So neurotransmitters are the chemical structures that release from the nerve endings. So these are just like hormones, but these are released from the nerve endings. So we can see in the diagram these now neurotransmitters which are stored in the vesicle in the nerve terminal, and the gap between the nerve fiber and the muscle fiber, this we call it as synaptic cleft. This is the gap, and the junction we call it as neuromuscular junction. If it is connecting with the nerve fiber, it is connecting with the muscle fiber, then we call it as neuromuscular junction. If it is communicating with the another neuron, then we call it as synapse. So we can see clearly that this is the motor end plate, the motor end plate. So presynaptic nerve fiber, and this is postsynaptic membrane, presynaptic membrane and postsynaptic membrane. In presynaptic membrane, uh, the vesicles, from the vesicles, the neurotransmitters were released, and it binds with the receptors present on the postsynaptic membrane. So with the help of another diagram, we can see. So this part is clear that the electrical impulses are traveling throughout the axon, and it is the nerve endings, the nerve terminal, the terminal buttons are stimulating the next fibers, right? So what happens? Specifically we see the nerve endings. What happens? So let us see with the help of this diagram. So when the action potential, this is action potential, is reached the terminal buttons, what happens? The permeability within the nerve fibers changes and causes and it causes opening of the calcium channels. Here in the yellow color channel, we can see these are calcium channels. So when the calcium channels are opened, so when the calcium channels are opened, what happens? There is a sudden influx of calcium ions, sudden influx of the calcium ions. We all know that calcium ions are abundantly present outside the cell membrane, and potassium ions are present inside the cell membrane excessively. The levels of calcium and potassium ions are more in the extracellular fluid, and the potassium ions in intracellular fluid. So what happens? See, this part actually what we are discussing is a presynaptic membrane, and this is postsynaptic membrane. On the postsynaptic membrane, we see the receptors, various receptors. These are all the receptors, various receptors. And the green color, these are vesicles, sorry, neurotransmitters, and which are stored in the vesicles. And these are the red dots, which indicates the calcium ions, which are in flux when the action potential reaches. So when the action potential reaches, the membrane permeability will change, and there will be a sudden opening of the calcium ions. So the calcium ion rushes into the terminal buttons, and the first thing what they will do is it binds with the vesicles. It binds with the vesicles which are present at the terminal buttons, and carry carry the vesicles to the surface, to the peripheral side, towards the surface, and the calcium ions break these vesicles, and causes release of neurotransmitters. So the condition when the calcium ions are not able to uh, uh, enter inside, that means when it is an autoimmune disorder in which uh, the autoimmune antibodies binds with these receptors, with these channels, sorry. So when the antibodies binds with these channels, it blocks the entry of calcium ions. This condition we call it as Lambert's-Eaton syndrome. Lambert's-Eaton syndrome. So depending upon the nerve fiber, the neurotransmitter changes. So in neuromuscular junction, the neurotransmitter is obviously, we say this is neuromuscular. The neuromuscular junction, it is acetylcholine. So this is acetylcholine. The neurotransmitter is acetylcholine. And so once the neurotransmitters are released, so the gap we are seeing here between the presynaptic terminal and postsynaptic terminal, the gap what we are seeing is synaptic cleft. So these neurotransmitters, say for example acetylcholine, releases into the synaptic cleft and binds with the receptors, acetylcholine nicotinic receptors. So when it binds with these postsynaptic membranes, when it binds to the postsynaptic membrane, what happens? Again, the postsynaptic membrane permeability will change, brain will become change. Then the membrane permeability will change and there will be a rapid entry of sodium ions. So up to this, we can say, up to this level, we can say there is excitation of the muscle fiber. So we can say this part, to this part, we can say it is excitation of the nerve fiber. What it is exciting? It is exciting a muscle fiber. So uh, the action potential when it reaches to the nerve terminal, it causes opening of the calcium channels. Calcium channels bind with the vesicles, and vesicles are moved toward the surface, and it causes release of the neurotransmitter. The neurotransmitter binds with the postsynaptic membrane, and it causes rapid influx of sodium ions. So in this picture also you can see that how this is connected, the neuromuscular junction. So how this nerve fiber is connected to a muscle, and these are the synaptic vesicles containing acetylcholine, and this gap what we are seeing is synaptic cleft. Synaptic cleft. So once there is a rapid entry of uh, sodium ions, rapid entry of sodium ions into the uh, sarcoplasmic reticulum. The sarcoplasmic reticulum mainly contains um, T tubules made up of C tubules and L tubules. So when these sodium ions get influx into the sarcoplasmic reticulum, was then the action potential against generated in the sarcoplasmic reticulum because of rapid entry of sodium ions. When there is a change in membrane permeability, so this causes this, in this picture we can see when the action potential, see here. So this is a T tubule. So when the action potential reaches, is traveling, and when it reaches to the triad in the sarcoplasmic reticulum, so first it will activates the dihydropyridine receptors. You can see here, this is dihydropyridine receptors. These dihydropyridine receptors activates the ryanodine receptors, which is present in the sarcoplasmic reticulum, and so once the DHP receptors are activated, these ryanodine receptors are pulled apart, in which the calcium is stored abundantly. So when this ryanodine receptor activated, the calcium is taken into the T tubules. The calcium is taken into the T tubules, and further it is utilized for the contraction of the muscle. So, so there are two receptors we must remember here. One is dihydropyridine receptors. So once this action potential reaches here, so this is in the resting phase. See, there is a closure of these ryanodine receptors and no activation of dihydropyridine receptors. Once the action potential reaches here, this causes activation of dihydropyridine receptors, and it causes further activation of activation of DHP receptors, the hydropyridine receptors. Then this in turn activates the ryanodine receptors for the release of calcium in terminal systems. So once the calcium is abundantly released, so who is stimulating all these things? Stimulated by action potential. So the action potential actually releasing this calcium within the sarcoplasmic reticulum. So once the calcium is released abundantly, then what happens? See, so the contractile proteins are troponin, tropomyosin. and actin filaments, myosin filaments, like that. See what happens. There are three components of troponins. So troponin T, troponin C, and troponin I. So this complete filament, we call it this violet colored, purple colored filaments. We see these are actin filaments, and these are lined by these actin filaments are lined by this straight line C. You can see this filament. We can say this one as tropomyosin, alpha tropomyosin, and the actin is made up of three components: troponin T. See, this is troponin T. Next, troponin C and troponin I. So during the time of relaxation, this troponin I, which inhibits the contraction, that is during the time of relaxation, the troponin I binds with the tropomyosin, right? Tropomyosin. And the active filaments, the active sites are covered. So few active sites in which head of the myosin will bind. So those active sites were covered when this troponin I binds with this tropomyosin until the calcium ions binds with the, see here, this is the first half of the diagram explains the relaxation phase in which the head of the myosin is not binds with the active sites because the calcium is not bind with the troponin C. So once the calcium binds with the troponin C, then only uh, this will display that active sites of the uh, actin filaments. So this is the first half. This is in the relaxed phase. During the relaxed phase, the head of the myosin is apart from this actin filament. So in the next half, we can see that the calcium ions, when it binds with the troponin C, what happens? Troponin C, the active sites of the actin filaments, the active sites of the actin filaments will be showed or will be displayed because the tropomyosin will move apart and it displays the active filaments of the actin filament of the actin. Then, so the head of the myosin, what happens when the active sites are active? That means whenever the calcium binds with the troponin C, the tropomyosin will be move apart, and the active filaments will be showed off. Then the head of the myosin utilizes the ATP energy and it binds with the active sites. It binds with the active sites and pull the actin filaments, pull the actin filaments and causes contraction of the sarcomere. So you can see there is a binding of this head of the myosin by utilizing energy. So ATP splits into ADP plus inorganic phosphate. Then it releases the energy, and the energy utilized by the head of the myosin filaments. Then it binds with the active sites, and initially it binds with 90 degrees side, and pulls it pulls the head of the myosin will pulls the um, actin filaments close together and causes contraction of the contraction of the actin filaments. So in this picture, you can see clearly, see in the first point it was given myosin heads are free from the actin filaments. It is not attached to the active sites. Then ATP splits into ADP plus pi, inorganic phosphate. Then the head of the myosin filaments becomes active, and it by utilizing energy, it binds with the active sites. So binds with the active sites. So this forms the cross bridges exactly at the 90 degrees. It is binding with the active sites of the actin filaments, then pulling apart. It loses the ADP here, ADP plus inorganic phosphate. Then it is, see here it is binding, and here in this step it is pulling inside. It is pulling inside. So this we call it as power stroke. The first point, this we call it as detachment. Second is hydrolysis. Third is cross bridges, and the fourth one is power stroke. Next, we can see here that the sarcomere, the length of sarcomere. So in this, we can say clearly the first part of this diagram explains that sarcomere is free. The relaxed phase of the muscle. See the length of the sarcomere. This is in the relaxed phase, and this is in the contracted phase. So side arrows were given to represent that there is a contraction of the muscle. Right. So once the head of the myosin activates, it binds with the actin filaments. When it binds the actin filaments, then this uh, causes contraction of the uh, sarcomere. Causes contraction of the sarcomere. Right. Then another important thing that I want to show you that the diagram, the video format in which you can see that an action potential when it reaching to a certain muscle fiber, it is spreading the impulses. So when it is spreading the impulses, there is excitation, contraction, coupling. So this is a sarcoplasmic reticulum. Right. This is a sarcolemma where the T tubules are visible clearly. Then, so the visibility is clear that these are all the T tubules in which the action potential reaches. To each and every part, the action potential reaches. So this is the whole sarcoplasmic reticulum. So this is the terminal system. Also, we call it as triads. That's T-tubules, and these are the two triads. And that we discussed. This is the dihydropyridine receptors, and the dihydropyridine receptors. Dihydropyridine receptors. The voltage sensitive protein. Dihydropyridine receptors open. The calcium channels will be released by activating the dihydropyridine receptors. Then the sarcoplasmic reticulum. Yes, we can see the excitation here, and it finally leads to contraction. Yes, we can see the contraction. So after this continuation of this one, this video. Yes. Now in this video, we can. These are the muscle fibers. These are the nerve terminals. which are acting on these neuromuscular junctions. The release of, yes, we can see the release of the neurotransmitter acetylcholine and binds with these. Yes, these are the nicotinic receptors. So after binding these nicotinic receptors, so we can correlate these action potentials also. Again, another action potential will be generated in the postsynaptic membrane. See what happens, there is opening of the sodium channels and causes influx of sodium ions into the muscle. So by that the positivity increases and there will be influx of potassium ions. See, we can say this is depolarization. And this event is spreads, this action potential spread actually throughout the sarcoplasmic reticulum, and these are T tubule, transverse tubule, the action potential which enters into the T tubule. We can see this type of this type of action potential is spreading everywhere in the sarcoplasmic reticulum. So once the action potential is spread, then what happens, see in this diagram. So yeah, from this see here, these are the actin filaments and these are the myosin filaments. And the calcium is released from the iodine receptor excessively. So this actually explains that when there is a release of calcium, there is a contraction of the circuit. When there is no calcium, there is a relaxation. Such a beautiful mechanism it was for excitation, contraction, coupling. So how these calcium ions are regulated? So these are the heads of myosin and tropomyosin, and these are actin filaments. See, when it binds with this, so the calcium binds with this, so it will be moved off. The active sites will be exposed. When the active sites of the actin filaments are exposed, it binds with the active sites. The head of the myosin binds with the active sites and try to pull the actin filaments inwards. So what is the, for the movement we require ATP. Sufficient amount of ATP is required for the movement of head of the myosin. It is utilizing the ATP for energy, and this we call it as power stroke. Exactly, this is called power stroke. Yes, you can see the video very clearly. It was attaching with the active sites and pulling it. So end of contraction. What happens when the end of contraction? When the calcium is taken up back by the sarcoplasmic reticulum, then it causes relaxation. This is called excitation, contraction, coupling. Thank you very much.